This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Thanks very much for joining. We're delighted to have Yuanting Zheng presenting today. He's doing his confirmation seminar. And just to give you a bit of a background on Will, as we call him, Will comes from a place called Heaven on Earth, which is the city of Hangzhou, which is about an hour out of Shanghai, a beautiful place by the West Lake. It's a very famous place in China, a very beautiful city. And he was fortunate to go to Zhejiang Agriculture and Forestry University, where he undertook a bachelor's degree in forestry, funny enough, from 2012 to 2016. After that, he did a master's at Zhejiang University in crop genetics and breeding over a period of 3 years. So he's done quite a lot of university stuff, and he saw the light and decided to come to Melbourne. So we're really delighted to have Will in our team working on aspects of parasites. And he's going to talk to you today about some of the progress that he's made in the first part of his project. So thanks, Will. Over to you.
Okay, thanks, Robin. I will share my screen. So can everybody see my screen?
Yep, we can all see it, thanks.
Okay, hello everyone. Thanks for joining my confirmation seminar today. I'm Yuanting Zheng, a PhD candidate major in veterinary science. My topic today is using advanced informatic methods to explore parasite genome and genomic dark matter to find new interventions. So before I talk, I would like to thank my supervisors, Professor Robin, Dr. Neil, Professor Johnny, and my committee chair, Professor Lee, for supporting and guiding me. Also thank the Australia government, Australia Research Council, and Yuanting for supporting my research projects. Firstly, I would like to introduce the background. As you know, parasites cause some of the world's most destructive disease, affecting more than 1.5 billion people and animals worldwide. In order to combat those diseases, researchers have developed many drugs and vaccines against those harmful parasites. Some parasites are well controlled by vaccines, but most parasites are only controlled by using chemical drugs. Recently, the overuse of chemical drugs has led to widespread drug resistance, which become a major challenge for controlling those parasites. This situation has stimulated us to search for novel drug targets and inhibitors. Well, there are many ways to conduct drug discovery, and one of the approaches is target-based drug discovery, which means that we will first detect the potential drug targets in parasites. Such drug targets could be a selective components in a critical molecular pathway. So it's very important to explore those molecular pathways in parasites. As you know, recently, big data has become a central part of biological research. Like omics technologies, including genomics, transcriptomics, and proteomics, have generated tons of valuable data, which accelerate the studies of those molecular pathways at different levels in parasites. Well, in addition, driven by rapid advances in laboratory techniques and equipment, we have the ability to explore specific molecular pathways and test the essentiality of pathway components in the parasites. So it's promising to conduct researches with those available resources. Then one of the big challenge for us now is how to effectively and precisely handle those big data. Fortunately, the development of machine learning methods enables researchers to integrate and interpret those big omics data. Successful examples such as AlphaFold2, which use a deep learning model to perform protein structural modeling, have made great progress. So together, combined with omics technologies and those advanced informatics methods, we believe now it's a good time to address some key biological questions in parasites, which can facilitate the discovery of novel drugs and interventions. Well, among all the parasites, I choose Haemonchus contortus as my research focus. Firstly, Haemonchus contortus is a socio-economically significant parasitic nematodes, which infects ruminants and such as sheep and goats, causing billions of dollars in losses worldwide. You can see the life cycle of Haemonchus contortus is rapid and has a number of key steps. First, adult females within the host animal lay eggs, and then the eggs are excreted in host feces. Individual first stage larvae hatch from individual eggs and commence the free living phase of the life cycle. Second and third stage larvae developed in the environment. Third stage larvae are the infective stage. Following ingestion by the host animal, we can see L3 exhales and developed to the fourth stage, and which then feed on blood and then developed to adult female and male worms. We can see from egg to L3 is the free living phase, and from the XL3 to adult is the parasitic phase. Well, secondly, based on the efforts of generations from the parasites community, Haemonchus contortus gradually become a model organism for parasitic nematodes, which means the experience gained on Haemonchus contortus could be applied to other related parasitic nematodes. Thirdly, there are extensive genomic, transcriptomic, proteomic, and lipidomic resources available for Haemonchus contortus, which is a good opportunity for conduct omics data-driven research on these parasites. So after doing a literature review, I found several knowledge gaps and formulate my research aims. First is the Ubiquitin proteome system. This system is responsible for the major protein degradation in eukaryotic cells, which is significant to protein turnover. Disruption of this pathway causes the accumulation of unwanted proteins and cell death. Although the Ubiquitin proteome system has been well studied in model organisms, there is scant information available for Haemonchus contortus. Because this system is critical, and many selective drugs have been designed and screened for potential inhibitors in this system for cancer, it's promising to explore the Ubiquitin proteome system in Haemonchus contortus. So my first aim is to explore this system in Haemonchus contortus. Secondly, although the genome of Haemonchus contortus has been assembled before and refined recently, there are still many unknown proteins inside its genome, which cannot be annotated by traditional sequence-based methods. such as BLAST. Recent developed structure prediction methods provide us an unprecedented opportunity to explore structure-based annotation. So I want to use various methods to annotate those unknown proteins and try to establish a refined workflow for protein annotation. So this is my second aim. Then, because we have plenty of omics data, I want to create or utilize a deep learning-based platforms to identify potential drug targets. This is aim 3. After that, I want to use laboratory-based methods to evaluate the accuracy of those predictions. So the first year, I focus on my first aim, the ubiquitin proteasome systems. After doing a literature review, I found that the ubiquitination pathway has been comprehensively explored in the free-living nematode, Caenorhabditis elegans, which could provide a good reference for us to construct the system model in Haemonchus contortus. So in detail, I used a homolog search strategy in this study. We firstly collected detailed information of the ubiquitination pathway, including genes, proteins, and families of domains in the extensively studied model organism C. elegans. Then we use various bioinformatic tools to identify homolog components in Haemonchus contortus based on their molecular homology. After that, we constructed the pathway and undertook a range of analyses, including gene-protein features, transcription patterns, protein structures, and a detailed comparison with the C. elegans pathway. Well, let us see the results. This is a summary of this pathway in Haemonchus contortus, which consists of 215 molecules. Firstly, ubiquitin is activated by the ubiquitin activating enzyme. Then the ubiquitin activating enzyme transfers the ubiquitin to the ubiquitin conjugating enzyme. After that, the ubiquitin conjugating enzyme binds to the ubiquitin ligase and transfer the ubiquitin to the substrate. Polyubiquitination of substrate results from rounds of contraction, and proteins, those substrate proteins labeled by different numbers of ubiquitin will later go to the proteasome to be degraded or take part in various cellular processes through proteasome-independent pathway. Finally, the ubiquitinating enzyme will cleave ubiquitin from the substrate, and those ubiquitins are recycled. Well, we investigate the transcription profiles of those identified genes across different developmental stages of Haemonchus contortus, including egg, first stage, second stage, third stage, fourth stage, and adult stage, male and female. We obtained a dynamic gene expression pattern and got some stage-specific and co-transcripted genes. This information provided a solid foundation for further exploration of this pathway in Haemonchus contortus. So after constructing this model, we want to know what parts of this pathway are conserved and which components are divergent, because this answer might help us to find the selective targets. So we compared the pathway with that of C. elegans at the protein level. We found that ubiquitin activating enzyme and ubiquitin conjugating enzyme are the relatively conserved components. particularly the ubiquitin molecule. Their protein sequence are nearly identical to each other, and all of these ubiquitin coding genes have higher transcription levels than other molecules in this pathway. We will particularly interested in variable components because we were searching for selective targets. We found marked differences in the number of ubiquitin ligase and deubiquitinating enzymes between Haemonchus contortus and C. elegans. In addition, we noticed substantial differences in their protein structure comparison. Those results indicate to us that there should be some ubiquitin ligase and deubiquitinating enzymes containing distinct binding sites for specific function during the ubiquitination process. Furthermore, we also observed differences between Haemonchus contortus and its host species in ubiquitin ligase and deubiquitinating enzymes. This suggests that some of those proteins might be selective drug targets. We will do further experiments to test them in the future. Well, in the chapter 2, I'm very excited to use our recently developed machine learning-based methods to model protein structures. As you can see, it's AlphaFold 2. This is an advanced, most reliable tool for our purpose. First, we want to know if there is such a protein whose sequence is very similar to its homolog proteins, but structural folding is affected by small differences in some key amino acids, result in huge differences in functions. We are also interested in orphan proteins, which could not find any homologs in any database at the primary sequence level, but matched with some similar tertiary structures of other known proteins in other species, especially those matched with catalytic sites of functional structure domains. We did not know the answer in the past, but we can do the test now with those advanced informatics. Then we applied these methods in this study. We predict protein structures and align them at the tertiary level. We obtain the conserved and variable regions and use that information to check some key catalytic sites of proteins and predict their functions in the ubiquitination pathway. It's very exciting to have those powerful tools now for us to study protein structure and apply them in our parasites research. In my following study chapters, I will do a structure-based drug screening on those proteins involved in the Haemonchus contortus ubiquitination pathway. So in chapter 2, we constructed here a model of the ubiquitination pathway of Haemonchus contortus. We explore the conserved and variable features and the transcription of genes in this pathway. We believe those results can contribute to the further exploration of novel drug discovery on selective molecular targets. Moreover, we propose using a machine learning-based methods to model protein structure, such as AlphaFold 2, which can largely assist our research in many aspects. For the following parts of my PhD project, I will extend the use of the protein structure modeling strategy to annotate the unknown proteins in Haemonchus contortus based on protein tertiary structure similarity. The three-dimensional structure of proteins contains a lot of functional information. So using advanced informatics technology to illuminate those structures can accelerate or can enhance our understanding of parasites at the molecular level. Chapter 2 addressed some knowledge gaps in M1. So I wrote my first paper, ubiquitination pathway model for the Barber's polyworm Haemonchus contortus, and published it in the International Journal of Parasitology, guided by my supervisors. In addition, I made a presentation at the Australia Society for Parasitology annual conference to share my research and also practice my presentation skills. Then, after chapter 2, I trying to work on another challenge, the functional annotation. As I pointed out in my literature review and the end of the chapter 2, there are still many unknown proteins in parasite genome which cannot be annotated only by sequence-based methods. Based on my experience gained from chapter 2, the structural modeling strategy performed by AlphaFold is very useful and might be useful for the structure-based annotation. Thus, I want to explore it further. So here I did a comprehensive literature review regarding methods and resources for protein functional annotation. As you can see, BLAST and Hammer search were the most widely used and representative programs for searching protein homologs based on sequence similarity. InterPNO-Scan and PFAM were platforms that could infer protein function based on protein domain information obtained from sequence features. Similar, like sequence-based methods, the structural search strategy was also developed for annotating proteins based on structure similarity. Here is an example, Cas gene 3Ds. Well, moreover, the PDB database contains all the experimental determined protein structures and their related function information, which means that we can predict the structure of unknown proteins and search the structure in the PDB database. If there is a hit with high confidence in some regions, then we can infer its function. Also, as you know, I think last week, Friday, the AlphaFold protein structure database announced that they had finished the prediction of 200 million proteins, a prediction of structure of 200 million proteins, which represent the entire protein universe. This will make a great impact on structure-based functional annotation. So having all those resources and tools available, I think it's a good time to improve the functional annotation of proteins. Here I use Haemonchus contortus as an example and to explore a refined annotation workflow. So this is my next chapter. We hypothesize that many proteins in the proteome of Haemonchus contortus, which cannot be annotated using a sequence-based methods, are annotable using a structure-based approach. Thus, we extend to constructed and critically evaluate an in silico workflow for the annotation of such protein, employing advanced informatics and the use of specialized database. As you can see from this workflow, I introduce it briefly. In step 1, we prepare high-quality protein sequence and structures from Haemonchus contortus proteome. And in step 2, we use both sequence-based and structure-based strategies to infer the function of proteins. We can also see that some methods are used machine learning model, and some methods just based on the sequence similarity and structure similarity. Then in step 3, we compare and integrate all the predicted function annotations. We believe after critical and comprehensive integration, we can reduce the unknown proteins in the Haemonchus contortus genomes and improve its annotation qualities. Currently, I have finished the step 1 and step 2. Now I'm pasting all the annotation results and compare them at different levels. This is my plan for my project. I'm currently moving forward according to the planned thesis structures. I expected that I can finish and address the 4 aims during my PhD. Also, next time we meet, I hope being able to tell you more about the advances I have made in this area. Thank you.
Thanks very much, Will. Fantastic. Are there any burning questions, comments? Please raise your virtual hand if you want to ask a question, and we can move ahead. So I've got an initial question, Will. I mean, what are the main limitations with AlphaFold, do you think, when it comes to predicting proteins and trying to use those models to interact in silico with drug structures, you know, with compound structures? Do you think there are limitations, or are you sure that they're going to perform well?
Thanks for your question. Yeah, I think this is also an issue that people talked it up a lot recently. From my perspective, I think there is a limitation for AlphaFold to predict the structures and then use this predicted structures to do in silicon drug discovery, because this method is still under development, and AlphaFold can only predict one structures for the proteins. But we all know that the structure of proteins have different phases. So use experimental methods can got different forms of the structures. That is a challenge for AlphaFold. But I see that they are the DeepMind groups. They are trying to improve the accuracy of AlphaFold. So I will keep pace with them and also test AlphaFold on different proteins to see which group proteins can be used in my annotation pipeline by AlphaFold, and which group of proteins still have limitation, and we will need other methods to figure it out.
Thanks, Will.
I've got a question. Yeah, thanks, Robin. Thanks, Will. Yeah, just on that last chapter when you're talking about looking at some of these targets in vivo, I just wondered if you could just expand on that a little bit in terms of what you have planned and how you might use that information to validate your methods, your in silico methods.
Thanks for your question. Yeah, I will use it. I will currently my plan is that if I can identify some drug targets, and then I will do a in silico prediction of the potential inhibitors which can bind the drug targets. Then some of them has been some of experiments has been done on other experimental animals. Then we can use their model, or we can buy the similar inhibitors or change some structures of those inhibitors to test it on our worms to test whether this is a real target or not.
No, that's great, Will. And I suppose what would you do if they don't work? You know, is there a way to go back and refine your in silico methods to, you know, do you think that information would be useful in terms of improving your in silico models?
Oh, thank you for your question. Yeah, this is also what I'm concerned. Actually, I think there should be some false positive results. Maybe the in silico prediction is perfect, but in reality, when we test potential inhibitors, there's nothing happen or don't have any efforts. Then, so that is very important that we explore at the molecular level, at different levels. We have omics, we have different omics data. If we have enough data to prove that there's a high probability that this target is a drug target, then I will go to test the inhibitors. And if there's no effects, which means there should be something that I ignored or I didn't find it, then this will be very interesting part that we can see what exactly happened to make these drug targets not works. Yes, I will expect it to see these results.
Any other questions, comments? If not, I'll Harry.
Hey, Will, thanks for the great talk. I really appreciate the style of presentation you give, quite straightforward and to the point. Just had a question about the transcription profiles for different ubiquitin proteins at different larval stages. I was just curious as to why you may think, or what reason that you get this generally across the board downregulation of ubiquitin proteins at the first stage larvae compared to the egg stage.
Actually, I think that the ubiquitin the ubiquitination pathway actually interacted with many other molecular pathways, and actually it takes part in various cellular process. So what we observed now is that the ubiquitin, the transcription of ubiquitin coding genes is higher than any other genes, but how it's involved into other process, we actually don't have much information. So it deserves to exploit at a molecular level. But this is what I observe now. Based on these results, it can support our hypothesis to exploit in further.
Thanks, Will. Thanks.
Thank you.
I think just Harry, extending that. So you're saying there was a down regulation of ubiquitin pathway components at L1. I mean, one thing to consider is that the parasite is exploding in terms of development and differentiation. So it's really growing, it's really adapting to the external environment. So there's really probably no point to degrade proteins because you probably need all of them as you're growing and developing to get through to the L2 stage, which is then going to start picking up. I mean, they're already picking up bacteria in the environment as they're growing, but they can't really, they can't really afford to lose proteins as they're growing through those first developmental There's also a massive upregulation in other genes as well during that period. The downregulation comes later overall of genes when you go from L2 to L3 to the arrested stage. That's when things slow down, and but again, that's probably linked to a downregulation as well, I'd say. But in the first phase, you wouldn't expect an upregulation because things haven't differentiated, tissues haven't developed and differentiated sufficiently. Once the adult has developed, that's when you'd probably expect more up and down regulation variation among tissues. But anyway, that sort of stuff is linked mainly to the biology of the parasite. But anyway, great answer, Will. Any other queries, questions? Housekeeping, Aaron?